Where all your tabs go, everywhere you go. There's no place like Chrome.
Let me start from the beginning, okay? Many years ago, I had a wonderful friend. You can probably see him in the emotes all around the stream. His name was Fish, okay? He was a beautiful, wonderful life partner pit bull that I got when I was in the market for getting a new dog, or getting a dog in general, for my first ever dog. And unfortunately, we had 5 wonderful years together, but he passed away in the beginning of COVID because he had cancer. He was a beautiful pit bull. Well, after that, I took some time to, recover and waited for, a couple of years now, almost on 3 years. Yeah, I told my community for a very long time that I was going to eventually get a new dog. I was eventually going to adopt a new puppy, and the time is now, folks. We're going to shelters today to kickstart the process. Now, let me explain something. Let me explain something. This is just starting the process. So I'm gonna lay down some ground shelters today to kickstart the process. Now, let me explain something. Let me explain something. This is just starting the process. So I'm going to lay down some ground rules. Some specific ground rules are I'm looking for certain breeds. Number 1 is a Bernese Mountain Dog. I think Bernese Mountain Dogs are very cute. I think they're very cool. I'm looking for a larger breed in general. I want a breed that I can wrestle with. You know what I mean? I'm looking for Bernese Mountain Dogs, or a Cane Corso, or maybe some kind of pit bull. There are some other breeds as well that are like almost impossible to find in the adoption in the shelter. Obviously, I will be adopting and not shopping. That's right. The puppy arc has officially begun. The time is now. Many of you already know that I used to have a wonderful furry little fellow by the name of Fish, and I will be getting a new puppy. I will be adopting a new puppy, hopefully today. In order to help me out, I have March right here, who's going to be manning the camera. Myung, coming hot off the heels of a 24-hour stream. She's basically delusional right now. Fusli, Leslie, who's here specifically for the love of the game. She has too many animals, and she might even adopt an animal. I think it's illegal for her to have more animals, though. Let's be real. She already has 2 cats. And last but not least, we have Val. Okay. All right. That's it. Let's get started. We're going to go inside. Dogs right in here. I think the dogs might be right. And the dog. That's a good. You're so right. Yeah.
Recording in progress.
They have iPads with dogs on it. You can just order a dog like a McDonald's kiosk.
Taylor.
There's a dog called Taylor.
The dog's name is Taylor. Look, it's like my shirt. How are you gonna name a dog Taylor?
Okay, so we can actually walk in and look at the kennels, but if we want like to basically hang out with a dog, if we find a dog that we really like, then we're gonna need someone to help us out. So in the meantime, I think what we should do is just like go through every single kennel, take a look, and get started, I guess. So you're a volunteer that works here?
No, I work here.
Oh, you work here full time. What is it? How does it feel to have the sickest job on the planet?
Awesome. I love doing it.
It's crazy. You get to play with dogs and cats.
Yeah.
And I think people should understand that if they're going to go out and adopt a dog. I want a puppy, obviously. I want a larger breed specifically, but if there are breeds that are available, the ones that I would like are obviously
Hello.
Bernese, Mountain Dogs.
Good afternoon.
Corsos, which I think.
Okay, let's get rolling so we can finish on time. Okay, so we're doing an activity session today. Student hours today and Thursday after class. Today, I can stick around for 15, 20 minutes or so, then I need to run to a meeting. Everyone has submitted their deep question 4 and is now doing the evaluation phase. Very good. So what we have, just a few weeks left. I think we have one deep question left. Which will not be this week, of course, because we just had one last week. Okay, so this week's learning objectives, learning outcomes. We're going to discuss genetic association and population studies, something called GWAS, genome-wide association studies. We're going to look at genetic variation in populations and so on and so forth. I think you guys all read that. If you did your pre-quiz, you're all ready to go. Okay, so let's look at one gene in a population. This is a population. It's not a balloon or something like that. It's a population, represents a population. How could we show there are multiple alleles for that one gene that we're looking at in this population? It's a population, and we're looking at one gene population. How can I show that there are multiple alleles? Any guesses? Okay, think about it like this. Is everyone in the population going to have the same gene? They're going to have that one gene that we're looking at. Okay, I got a few people that are awake here. Yeah. Talk a little bit louder. Old guy here, don't hear so good. I'll come to you so I can hear good. Okay, say again. Yeah.
Bro, you and Albert are playing solo.
Yeah, but so the question, guess my mic does it work? Yeah. The question is how can we show that there are multiple alleles of that one gene? Okay, so we're looking at a population is that circular, right? And is everyone in the population going to have that one gene? We all have the same genes, right? So everybody in the population is going to have the one gene. And so if I wanted to show that there are different alleles, it's all going to add up to 100%, right? If I look at it in a bubble. So here it'd be like, there's one allele. How would I break this down into,
I accidentally
do a pie chart.
Instead of request.
Awesome, that's what I was looking for. Gonna do a pie chart. Yes. Okay, so if we wanted to show multiple alleles, this is looking at one gene and we're gonna show multiple alleles, it would make a pie chart. We make the pie slices different colors because it adds up to 100. Everybody in the population has the gene. The pie slices are gonna add up to 100%. And then it'd be like what proportion of the population has a particular allele. So it maybe make like one of the wedges yellow, one of the wedges blue, and so on. Yeah, very good. Excellent. All right, so we're on track there. Now, okay, so what does this mean? Again, I'm looking at one gene, and it says here, one species in 3 populations. What do the pictures mean?
Yeah.
How many alleles we got here? What's going on with the populations? Yeah. Yeah, one gene, still looking at the same gene, and different colors of circles. So I guess different alleles, 3 different alleles. And it's showing us, well, population 1, I don't know what the population is, it only has the 1 allele, and then population 2 has another allele, population 3 has a 3rd allele. Okay, so each population only has its 1 allele, very different from one another.
Bro, the thing is,
Not all populations have the same allele. All right.
What
How
do you
about
do if you
this?
max it out? Can't get any more.
Now we got to what you were talking about. What do we got going on here? 3 populations, 1 gene of interest. So each of the populations, there's only one, we're only talking about 1 gene. What do these mean? What does this mean now? Yeah, you might as well. Bam, nailed it. 1 gene. And there's 5 alleles. 1, 2, 3, 4, 5 different colors of pie slices. Those are 5 different alleles of the same gene. So, and it's showing you like the proportion of those alleles in population 1, and then the proportion in population 2, population 3. And so the red line I drew in there, that's actually the same allele. I'm just emphasizing it's the same allele, and its proportion in the various populations. They look about the same. Okay. Does everybody follow so far? You want to be following up to this point. If you're lost now, oy. Okay. Any questions? Okay, good. All right, so in groups of 2 or 3, you have 2 categories of variation exist. Okay, variation within a group, or a population within a group or a population, versus variation between groups or populations. Considering just 1 gene, which of the situations below, A or B, shows greater within group variation, and which group shows, let's do the first one, which group shows greater within group variation? And you know what? I want to see. I know Christian, my man, he's on it. But I want to know about the 99 other students that showed up today. We will discuss. We're going to discuss. Okay, bing, I just sent you a clicker, just in case you weren't, you're on it there. A or B? So again, we're going to go with the first question first. Which of the situations below shows greater within group variation? Yes, a question. Yes, were you? Oh, you were giving the answer. Oh, I'm sorry. I thought you had a question. Okay. Yes, hold it. Hold on. Don't give away the answers just yet. Okay, 5, 4, 3, 2, 1. I just want to say, wow, 89%. All right. and people are actually cognizant we're here because nobody picks C, D, or E. Excellent. So 11% are saying the answer is B. 89% are saying A. Okay, so that was which of the situations below shows greater within group variation? Within group variation. Okay, so most of you guys, 89% are saying A. All right. Which shows greater between group variation? Between group. Five, 4, 3, 2, 1. Oh, I like this. So you guys are actually really, you're paying attention very well. So it's the same proportions. People are answering, so your guys are paying attention. So you're saying within group, majority of the class said within group. This over here, this is showing the greater within group variation. And when I said between group variation, most people are saying 82% are saying greater between group variation is being shown over here. All right.
I got you.
So I guess every time you see one of these pie charts like this with alleles, you're going to want to ask yourself that question by seeing greater within group or between group variation. So the answer is, and yeah, we can come back. Now can we have the answer? Maybe not at the, yes, can I come back to you now? Because I want you to say your answer you're saying. Just yell it out. Oh, you're changing your answer? There's discussion. Huh?
You should have got A.
Within group. A. You said A. Okay, so most of the class said A, and A is the correct answer. So which of these here, A or B, is showing greater within group variation? Hey, look at this. If I were to take two people and I were to pick, I'm going to just take two people randomly out of this population shown here. What are the chances that I'm going to get individuals that have the orange allele? Huh? I'm choosing them from this right here, this group right here. Very low. Okay, well, let's try again. No, that's okay, that's okay. So I would get them, right? There's no variation. I pull those people out randomly. Okay, so this particular group, there's no variation. As with this group, there is no variation. Now, if I were to randomly pick two people out of this group right here, what are the chances that I'm going to get two people with the orange allele? It's not so likely, right? It's a 1 in 5 chance each time I pick. 1 in 25 total. Anyways, and so this within group variation, since I can pick randomly two people out, I'm actually likely going to get a lot of variation between my picks. So that's one way to help you think about it if you're confused, right? So there's higher within group variation for the groups in A, as there are B. There is no within group variation. There's actually only between group variation, right? There's only the variation between the groups. Okay. Okay, so some people think there are genetic differences between races of individuals. That race is a biological, not a social construct. Which of the above sets of graphs would support this hypothesis? Again, I'm not saying this is true. I'm not saying it's false. I'm not saying anything. I'm just asking if these people that came along and said, so there's genetic differences between races of individuals, think that there are different oh my gosh, I'm losing my. Which of the following graphs supports the hypothesis that there are genetic differences between races? Yeah. It would be set B. Very good. Right? If that would be, so because we got these different groups here, and the groups, this group uniformly has one allele, this group has a different allele, and this group over here has a different allele. Now, some people think that this is so. They think that there are genetic differences between races. And you notice how it says Africa and Europe. These are actually locations, right? They're pretty much very, very large locations. But some people, again, these are locations that have been, what's the word? It's been implicated that they're representative of particular races, right? Now, again, I'm not saying that. That's just what has been said. So Africa versus Europe versus Middle East. Okay, so below are data from a study, Rosenberg, 2011. This is real data, okay? Does it support or refute the claim that there are genetic differences between different races of individuals? So again, these are locations, right? Africa, Europe, Middle East, they're locations. They're often stigmatized as being representative of specific races. I'm not saying that they are, but I'm just going to say that this is an example, okay? Okay, so A, yes, it does support that there are differences. And B, no, it doesn't support that these are differences. Okay, 5, 4, 3, 2, 1. Wow. Oh. Okay. We are essentially 50-50 split between A and B. Okay. So in groups of 2 or 3, we have 2 categories of variations that exist here, A and B. Okay. 2 categories of variation exist. Variation within a group or population versus variation between groups of populations. Considering just one gene, which of the situations A or B below shows the greater within group variation, the greater between group variation? So you actually already answered this for me, right? You already told me, well, A has greater within group variation and B has greater between group variation. You already told me that. So rather than discussion, let's just move forward. So some people think that there are genetic differences between different races of individuals, i.e. that the race is a biological construct rather than just being a social construct. If that were the case, which set of data, set A or set B would be true? If it were the case that there are genetic differences that define races, would it be set A or would it be set B? I'm not going to do the clicker. I want actually to. Yes. It'd be set B. Does everyone agree about that? Okay. If indeed these people, I don't know who they are correct, that races can be characterized, they have genetic differences, it would be set B. We would expect to see for this particular gene Africa to have a different allele than Europe, and then the Middle East, and so on and so forth. This was the real data from Rosenberg. So, what does it support? Does it support that there are indeed races have are represented by genetic differences? So, in other words, is there more differences between these groups, or if you were to pick two random people within a group, would you expect to find more differences within a group? Okay, I'm going to poll you on this. Does the data support, yes, that these are different? There are genetic differences between races, or does it not support that? 5, 4, 3, 2, 1. Wow. I'm confused. I'm confused, but okay, so only I'm confused because I thought it would be 100%. I thought you guys might be 100% after having a discussion, but I guess there's some confusion because only 60% of the class are saying B. No, it doesn't support that these are different. If all of Africa was one color allele, let's say red, and Europe was all blue, and Middle East was all yellow, wouldn't the data support that there are distinct genetic differences between races? That's not what we found. 60% of the class, 100% of the class should be saying, no, it doesn't support that. It actually looks to me like there is more genetic variation within any one of these particular populations. If you were to randomly choose two individuals, randomly choose two individuals from Africa, what are the chances that their genotypes are going to be the same? Essentially not very high, right? From Europe, randomly choose two individuals. What are the chances that they're... So, in other words, there's more within group variation in each of these populations, okay, than there is between groups. So, think about that one. Okay, so, it doesn't support this notion that race is a biological concept. That's actually nonsense, and there's actually much more genetic variation within a race than actually we find between races. If you were trying to explain why you are so different from other humans, then it is more likely the reason is due to within population variation. If you took an individual from any of these populations, could you determine what population they came from based upon their alleles? Yeah. Did everyone hear that? So no, it'd be very unlikely that you would actually guess correct what population that this person came from, because there's so much within group variation, you wouldn't know. Actually, they all kind of look very similar in terms of their distribution of the overall alleles, right? Okay, so no, you couldn't. There's no biological basis or genetic basis for the term race. It's kind of an obscure thing, right? Doesn't mean racism isn't real, and it doesn't mean that race isn't something that can impact people's experiences and opportunities and so on in a social context. Okay, that's not what we're saying here. Okay, what does it mean if you randomly pick two individuals from the same ancestral group? What does it mean if we pick two individuals? You pick two individuals. If you randomly pick two individuals from different groups, you're likely not going to get two individuals that have the same alleles, right? And sample 2 people, sample 100 people, you're going to get a lot of variation between them. So take a look at your neighbor sitting beside you. Your DNA is 99.9%. 99.9% of the nucleotides in your DNA are identical to whatever neighbor you just looked at. Okay? And, but you have 3 billion nucleotides in your genome. That leaves only 0.1%. What is 0.1% of 3 billion? That's 3 million loci where differences can occur. That's a lot of differences, right? It's a lot of differences, but in terms of the overall composition of our genomes, it's extremely small. There's not a lot of variation between us. Those differences, what are they? They're SNPs, they're microsatellites, they're repetitive elements. We discussed all these before, right? And if you were to choose any of the above populations, if you were to choose any of those, so 95% of the 0.1% difference that is possible is found to 95% of that 0.1% that can be different among us. 95% is represented within group variation. Okay, 95%. Only 5%. Only 5% of that 0.1% is actually representative between group variations. It's extremely, extremely small. Okay, so this is just a quote, we are alike in the ways that we are different. Note that, what does this mean? We are alike in the ways that, well, you can think about it. Okay, so how do we know that genetics is involved in producing a trait? Genetics is involved in producing a phenotype. How do we know this? We talked before about how environment can very much influence our phenotypes, right? So how do we know that genetics is involved? And there's, by doing the various studies, twin studies are an example. So you're familiar with monozygotic twins, identical twins, right? Monozygotic, these twins arise from a single fertilized egg cell. They have identical DNA. The zygote splits and into two very early in development, leading to the formation of separate embryos. So they have the same genotype. So people study like, okay, well, what exactly is the contribution? The twins are growing up, they grow up in different environments and stuff like that. So what is the contribution of genetics versus what is the contribution of the environment? Dizygotic twins, on the other hand, they result from fertilization of two separate eggs. They have two different sperms during pregnancies that are very different. But anyways, so twin studies help us understand how genetics contributes to traits. We also, the lab studies where you knock a gene out and you put a gene in and these kinds of things. inheritance patterns. So you've all seen pedigrees. You've probably even been asked some pretty difficult questions about pedigrees in your previous genetic courses. What is it? Sickle cell anemia and the bloodlines and all this. So that helps us understand genetics. Then we have population studies, but it can be very difficult to rule out environmental effects. So, thinking about it in terms of like genetics and heritability and environment, what can heritability tell us about a trait? Okay, 5, 4, 3, 2, 1. 54% say C, 38% A. Only 7% say B. All right. We'll come back. Heritability, trait heritability. So we're talking about traits and inheriting traits. So the proportion of variation in a trait in a population, what proportion is actually due to genetic variation? And that's heritability. So heritability equals the amount of variation explained by genetics divided by the total phenotypic variation. It's hard, like I said, it's hard to study traits. What do we normally do? We do some tricks. We'll put the organism in an environment. We control the environment. And then, in other words, we're removing that environmental effect. And so we can study just the trait, like when we have a mutation in the trait, and how is that a mutation in the gene? How is it going to affect the trait? So that's kind of like a trick. But then we often need to go to do field studies, right? Where we look out in nature, or even if you look into clinical studies and so on with humans, you can't put humans in a controlled environment, for example. So you can study the amount of variation that's explained by genetics versus the total amount of variation that you find in the population. And so their heritability ranges from 0 to 1. 0 would mean that genetics doesn't explain anything about the variation that's seen in the trait. Note how I'm saying variation of a trait. Whereas all the way up to 1, genetics explains everything about that trait. So if you had a particular allele or a set of alleles, you're bound to have a particular trait, a particular phenotype, right? And so this is like a heritability, that's calculation of heritability. And so you can see here that height is only, it's 0.8. So it's only 80%, 0.8 in terms of the heritability scale, explained by genetics. And 20% is not. Okay, so you may have individuals in your family, like maybe like your grandparents, much shorter than you. And why does grandpa he's much shorter than I am, and maybe grew up at a time in a place where food wasn't readily available as it was, or was exposed to chemicals and so on. So there can be an impact, but 80% of the height should be determined by heritability, so by genetics. Left-hand preference, 0.25%. So that's actually quite low. It's quite low. It's more of a learned thing, right? And then eye color, eye color, 0.61 to 1%. Okay, so heritability, explaining, how much does genetics explain a particular phenotype? And that can vary with age. So if you look at this, just looking at the gray bars, never mind the red ones made now, you can see for this particular trait, this is a Nordic twin study of cancer. They're looking at variation in liability. So kind of like your risk to get cancer, let's say. And in the twin of studies, 315,000 twins they looked at. And they found that it was genetics, so heritability could explain up to, what is this, 0.5 or so, 55, so 0.55% on the heritability scale for people at age 40. So whether they're going to get cancer at age 40, right? But as you get older, now all of a sudden, heritability doesn't really explain it anymore. So this is really unfortunate. It happened in my family. People as they get older, your likelihood of getting cancer, it gets higher, right? And so if my grandmother at, I think in her late, maybe 68 or 69 years old, found out that she had bladder cancer, right? And so my mom's all scared. Oh my goodness, genetics, it's a heritable thing, and she's terrified. Am I going to get this and everything? But grandmother is already 68 years old, and at that age, if you get cancer, it's not so likely that it's explained by genetics. It's actually you've acquired mutations throughout your lifetime. They've kind of built up, be exposed to various insults and environments, your cells, and so now at that age, it's just kind of like accumulation of mistakes. Your probability of getting cancer just increases, and so heritability has less of an explanation. You can see that it only explains 20 to 25% after the age of 55 that you will actually get cancer from inheritance. Does that make sense? Yes? No? Yeah. Okay. Oh, yeah. So I hope that makes sense. Like as you get older, now all of a sudden your likelihood of getting cancer, it's less so explained by genetics and just more so explained by the environment. We want to study which genetic variants contribute to variation in a trait. We need which of the following? You want to study like how much genetics contributes to cancer. Okay, which one of these do we need? 5, 4, 3, 2. Okay, so C was our highest guess, only 51% though. And then B is 35, A is 14. All right. Okay, so remember whenever we looked at the eyes, right? We were looking at the eye, and I don't know if you remember, but we were saying like in the population, in individuals, actually this anterior border layer, there's gonna be a lot of variation in pigmentation. Okay, a lot of variation between people. And then we talked about this particular layer down here, and we were saying like there's almost no variation unless you're an albino, you have albinism and you can't produce pigmentation. This is going to be pigmented, and it's pigmented for everyone. Okay, so we talked about that, right? So if we were looking at genetic variation, just imagine these two scenarios. So like we're trying to figure out eye pigmentation, and if pigmentation didn't change, and we were looking at all kinds of genotypes, right? All kinds of alleles. If the pigmentation didn't change, would we be able to know what alleles were contributing pigmentation? Well, if it's not changing and there's no variation, how do you do that, right? All right. If we want to study which genetic variants contribute to variation of a trait, we need, which of the following do we need? We need which of the following? Well, 5, 4, 3, 2, 1. Okay, it's starting. So 77% are saying B. Very good. I think you guys are thinking about this. You see it. I didn't tell you. You thought about it. You came to the conclusion. 77% said B. Both, you need to have both genetic and phenotypic variation between individuals. If I got all kinds of different alleles and they all have the same eye color, well, you know, how are you going to figure out which alleles contribute to eye color if you only have one eye color? But if I got a whole bunch of individuals, different alleles, and they have different eye colors, well, now I can start to look for an association, a correlation between certain alleles and certain eye colors, right? So you need to have both the phenotypic variation and the genotypic variation. Okay, so whenever we're looking for loci, we're looking for genes that are associated, their alleles are associated with particular phenotypes. It's this is a concept I need to point out. It's much easier if the genetic variants have a large impact on the phenotype. Okay, so that makes everything easier, right? In other words, if I was looking at all the different alleles in the genome and I was looking at all different eye colors, and actually I have this one allele that I found that 80% of the time, that allele is particular allele of that gene is going to correlate, it's going to be inherited with a particular eye color. Okay, that's 80%. That's a huge heritability, right? It's a 0.8% heritability, very high. So traits that have this, that have major contributors, so genes that are alleles that are contributing mostly to the heritability, are easier to identify rather than traits that are comprised of 50 genes and they all contribute only 2% of the variation. So you imagine have all 50 different genes in their alleles, and each of those genes is only contributing 2% towards that final phenotype. Very difficult to identify those genes. And so you know that whenever we talk about Mendel and the traditional work and all that, like they were choosing traits where the alleles had a major effect on the phenotype. They unknowingly so or knowingly so, but they were able to focus in on those alleles. Okay, so larger sample size is needed if a variant has a small effect. So you could still identify genes. We're going to talk about GWAS and identifying genes using variation in populations, but you can still identify those genes that have a small contribution if you look at a very large population. It's a statistical thing. Okay, so in groups of 2 or 3, you are investigating genetic associations with cardiovascular disease. Again, the cardiovascular. How would you ensure you're including the full phenotypic variation or genotypic variation? Okay, so discuss. how would you ensure you're including the full phenotypic variation or genotypic variation? Okay, all right. So, what conclusions did we come to? Oh, we're still figuring it out. Hey, we still got an activity to do, so we should move quickly on this. So anyone want to tell me how would you ensure that you're including the full phenotypic variation? Hey,
That increase the chances that you have more variation for that core cardiovascular disease among the other.
yeah, so Christian's saying you got to try to include as many people as possible with cardiovascular disease and right, for their phenotypic part, right? As many as possible, yeah. But as many as possible that have cardiovascular disease and that don't have cardiovascular disease. Try to go as wide, cast as wide a net as possible. Absolutely, right? And we're going to try to, as wide a net as possible, as many people as possible. We're going to try to get as much variation as possible in the phenotype and as much genetic variation as possible. And I know you're still thinking, well, how do then what do we do? And we'll talk about that in a second. But yeah, okay, great. And I just want to reiterate, though, it's really important to, we talked about this before, to define your phenotype. Okay, so you have a particular phenotype. You have to define it very, very accurately, because what's cardiovascular disease to one person might be completely different to another person. And so, how are you going to, this can be for continuous trait or whatever, but if it's continuous, you have to maybe set a threshold of something, I don't know, blood pressure, whatever. But you have to define your phenotypes very, very carefully. Okay. Right? Okay, so we're going to talk about GWAS. We're going to talk about how that's done. So GWAS, essentially, genome-wide association studies. We use a lot of molecular markers. So molecular markers, we talked about those before. They provide useful landmarks for genetic mapping. Okay, so it shows the chromosome, and at various sites, there's got these flags. I guess that's supposed to represent the markers. But okay, so remember, and we're talking about trying to find correlations between certain genetic loci, so certain alleles, genetic variants. Variance is the key word. So you need to have sites, loci, that have different alleles. That's going to make a great marker. If those are known in the population, which in humans, they're very well known, all these different loci. So all of these SNPs, RFLPs, microsatellites, we talked about those before. And those are all very useful to serve as markers. Now, okay, so alleles for genes located close to each other tend to be inherited together. I want you to realize something. Every year I say this, and every year I think people, there's a portion of the class that just And I'll reiterate it. I don't know what's happening there, guys, but okay, the actual SNPs that we're going to be following, those different heritable variants, okay, if you find that, okay, 80% of the time, this particular SNP, this location, this A is inherited with blue eye color or whatever, it doesn't mean that that SNP, that marker actually is causing the blue eye color. Nice. I can almost count the people in the class that are going to get this right on the deep question. I have an estimation. I'm locking it in. Okay. Now, doesn't mean that it's going to be the causal agent of the particular trait that it was associating with. It means that it is likely that its correlation, since it is correlated, it's linked to this particular trait, that something inherited nearby might be that allele itself, that polymorphism itself. It could be a neighboring allele that's actually following in inheritance that particular marker, a nearby gene that's responsible. Okay, just putting it out there. And we looked at haplotypes, and so haplotypes, great, so sets of linked alleles and everything, and so it's just important to distinguish. They're not necessarily the causal agent. So what, you remember what SNPs are? You know, you have all these variants. It's a particular location. You know, here we're lining them up, and I guess we have different individuals, and this person has an A, that person has a C, and this person has a G, and so on, at that particular site. Okay, so those are SNPs. You need to have 2 or more versions in order for them to be of any use. You need to have variation. That's polymorphism. That suggests there is variation. In order for them to be useful, to trace their inheritance pattern with the trait of interest. Okay, and so they must be present at greater than 1% of the population. It's kind of semantics. Below that, they're considered a mutation. But anyways, so it's just generally they're present at the higher, they got to be present in order for you to have any statistical significance. All right, so GWAS, what GWAS does is it identifies regions of the genome that are associated with a phenotype of interest. It can be a disease, it can be eye color, it can be height, whatever. You define your phenotypes, okay? You have controls that, you got to draw, it can be continuous traits, so it can be height, different statistics, or it can be yes or no. Has cancer, doesn't have cancer, that kind of thing. And then what you're going to do is you're going to, you take your DNA from an individual and you look on your SNP, you transfer to a SNP array, and it's going to, you have all the SNPs all across the entire genome, and you would use statistics. You're not going to do the statistics, they're very complicated, but you would determine whether there's a correlation, whether a particular SNP is being inherited at a high frequency or not with your trait. It helps find genetic causes of particular traits. That's what GWAS does. Okay, so this is this is called the Manhattan plot, and this is, essentially how GWAS works. All of these dots are different SNPs. And you can see here that each dot is a different SNP, and this is their position on the chromosome. Chromosome 1, chromosome 2, chromosome 3. It's gone to one end to the other along the chromosome. And what's measured on your y-axis here is the probability. It's like how do I describe that? The likelihood, it's the correlation of your SNP being inherited with your particular trait. So I don't know what trait they're measuring here. It doesn't say. But anyway, so the higher that the dot is on this graph, that log 10 p-value, why do we not use p-value, just a regular p-value? Like it's measuring correlation, right? If we measure regular p-value, it would just, everything would be above our threshold line here. Everything would be correlated. So we really knock down, without getting technical, we knock down the p-values here. So something has to have an extremely strong correlation with your particular trait in order to be high up on this graph. And the higher the dots are on this graph, the greater the correlation they have with your particular trait of interest. And so you can draw a threshold like they've done here, or you could just say, well, I know this one here is much higher than these, so it's more likely to be contributing to the trait than those ones. Once you find your dots, you found the SNP, you found the allele, because all the alleles are represented here as well. You find the allele that's associated with your particular trait, okay? But again, you don't know whether the SNP is causative or not. You would have to do further testing to figure that out. Okay, so how much time we got here? Yeah, I think I can give you 15 minutes. I'm going to open this up. You got an activity to try. Just try to do as much as you can. And, it's only, you have to submit it by Wednesday, right? So let me go and open this up. Okay, it should be visible now. Week 10 worksheet. I'll give you 15 minutes.
What the fuck? This nigga's retarded.
Two minutes left. Again, if you don't finish, you have until Wednesday. Maybe just find a spot outside the class. 2 minutes. Okay. Okay, just try to finish it, when you can. Let's do Wednesday. Make sure you add your group members' names when you're handing it in. Do Wednesday. Okay, so in the last, 2 minutes we have here. Why would an SNP be associated with a trait like fur color? Yeah. Yes. Yes.
Another reason that it's actually part of the population is that it's a part of the
Very good. That's the key concept I'm hoping everyone is getting out of this particular activity. Christian was saying that these SNPs that we're finding here, why would they be associated with a trait like fur color, for example? The answer is that they could be a particular SNP is linked to the causative agent nearby gene or whatever it is that's contributing to fur color. It's not on the allele, the GWAS mapping itself, but it's actually a difference that's nearby that particular marker. And the other thing could be is that the SNP is actually part of a regulatory sequence, causes a difference of expression of a nearby gene, or it could be part of a coding region of a gene. Okay, so even outside of a gene, it might be linked. Okay, so when you look through your SNPs and you're looking at fur color, you likely found some that had complete association with fur color, such as this one here. And other random ones had no association whatsoever. They're completely unassociated with fur color. Okay, that's how GWAS mapping works, is you look for the ones that are associated with a particular trait, but it's just a starting point. It's not telling you it's for sure the causative agent of whatever the thing is. Okay, thanks a lot, guys. I'll see you next class.